In this video, we are going to look in detail at the process of gluconeogenesis. So we have already discussed that we can obtain glucose from our diet. We can obtain glucose from breaking down glycogen. And now we are going to be discussing a process in which glucose is going to be made from pyruvate by reversing the steps that we already discussed in glycolysis. And you may ask yourself, why is it that the cell has all these different ways to make glucose? What is the need for that? Well, to be honest with you, if we think about it, there are going to be instances in which the cell has to maintain blood glucose levels. During periods of fasting, starvation, low carbohydrate diets, and even intense exercise, the body needs to find a way to maintain glucose levels. Understand that gluconeogenesis is a process that is highly endergonic. So what that means is that it requires energy to convert pyruvate all the way to glucose. So one of the things that we should establish now is that pyruvate to glucose is going to use many of the reactions that were utilized during the process of glycolysis to convert glucose to pyruvate. In the case of gluconeogenesis, again, we are starting at the level of pyruvate and we are going to be synthesizing glucose. Now, gluconeogenesis have different starting points, and the sources of molecules in this pathway can come from a diverse resource of molecules that are obtained in different pathways. So, I'm going to now outline different resources for molecules involved in gluconeogenesis. So gluconeogenesis can be obtained from lactate being converted into pyruvate. We can have keto acids that get into the citric acid cycle, obtain oxaloacetate, and then make phosphoenolpyruvate. We can also have glycerol, and what we're going to see in chapter 21 is that glycerol, when you hydrolyze triglycerides, is one of the products. So glycerol can also be converted into a molecule that is called dihydroxyacetone phosphate, which we have seen previously, and that can enter gluconeogenesis in order to synthesize the wanted glucose molecules. Now, if we look directly at gluconeogenesis, the difference between gluconeogenesis and glycolysis is at the following steps. The first difference between the two processes is at the first step. So remember, gluconeogenesis is going to be the reverse reaction, okay, from glycolysis. And in the last step of glycolysis, which technically is going to be the first step in gluconeogenesis, we had phosphoenolpyruvate being converted into pyruvate. So logical sense means that pyruvate needs to be converted into phosphoenolpyruvate in the process of gluconeogenesis. But interestingly, this is not a one-step reaction. The conversion of pyruvate to phosphoenolpyruvate requires several steps and several enzymes, okay? After we have converted pyruvate to phosphoenolpyruvate, and we will be looking at those specific reactions more in detail in sequential slides, understand that the next few steps are the exact reactions that we saw in glycolysis, but then in the reverse form. Then the second step that is different between gluconeogenesis and glycolysis, as you can see, is the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. Understand that in this case, because we need to remove a phosphate, we need to utilize a phosphatase, which is a hydrolase enzyme. And then the last step that is different between glycolysis and gluconeogenesis will be the formation of glucose from glucose 6-phosphate. In this case, we also need a phosphatate enzyme to hydrolyze out the phosphate group to generate glucose. Now, understand that specifically, and we observed this previously, glucose 6-phosphatase, it is actually present in the ER. Now let's have a closer look at these three reactions that are different between gluconeogenesis and glycolysis. So remember, when we are trying to convert pyruvate to phosphoenolpyruvate in the process of gluconeogenesis, there is going to be several steps that we have to go through. So first, pyruvate is going to be converted to oxaloacetate. And as you can see, in this process, ATP is hydrolyzed in this reaction. So the energy that is needed in this endergonic process is going to be coming from the hydrolysis of ATP. Understand that the enzyme that catalyzes this reaction is going to be pyruvate carboxylase, and this enzyme has multi-units because there's many things going on. We are going to be increasing the number of carbon atoms. That's where the CO2 comes from. We are also going to be hydrolyzing ATP to ADP. Okay, and as you can see, even water is involved in the formation of the oxaloacetate. So once this oxaloacetate is processed, remember, pyruvate is going to enter the mitochondria, convert itself to oxaloacetate, then oxaloacetate will be converted to malate. Malate will be shuttled into the cytosol to then form oxaloacetate again, and then we are going to be shuttling it through gluconeogenesis. Now, understand that the oxaloacetate must be transported, okay, from the mitochondria in the form of malate. Malate, understand that it is the molecule that can be transported through the mitochondrial membrane and then is converted back to oxaloacetate. So you may be asking yourself, why is it that we need this molecule? If we remember from the process of glycolysis, we are actually going to be in the process generating NADH, okay? And one of the things is that in this process of conversion from pyruvate to oxaloacetate that needs to be shuttled through the mitochondria, this step of the reaction is also going to be producing NADH. And that NADH can be used in further reactions. Then, understand that that oxaloacetate then is going to be converted to phosphoenolpyruvate. So notice that the conversion of pyruvate to phosphoenolpyruvate takes 2 steps. The second enzyme involved in the process is going to be carboxykinase, specifically phosphoenolpyruvate carboxykinase. So this carboxykinase, as you see, is going to be utilizing GTP in the reaction to form GDP. Overall, in the reaction, we are going to be removing a CO2. But understand that here, because GTP in a way is similar to ATP, this reaction is going to be the driving force, meaning the energy that is going to be required in order for this endergonic process to occur. So again, as you can see, it takes 2 steps to go from pyruvate to phosphoenolpyruvate in the process of gluconeogenesis. Then there's 2 more reactions that they're different from one each other. And these 2 reactions are going to be hydrolysis reactions. Once the phosphoenolpyruvate gets all the way to the fructose 1,6-bisphosphate, as you can see, we need to convert fructose 1,6-bisphosphate to fructose 6-phosphate. Because we need to remove a phosphate, we are going to be utilizing a phosphatase enzyme, meaning a hydrolase, to remove that phosphate from our substrate. The other hydrolysis is going to be occurring specifically in the endoplasmic reticulum. So understand that glucose 6-phosphate, it needs to be converted into glucose in order to be utilized. Again, that glucose is going to be shuttled back into the bloodstream. And again, we are going to be using glucose 6-phosphatase, which is an enzyme that we already saw in the breakdown of glycogen. Now it is also involved in gluconeogenesis, and this is how we go from glucose 6-phosphate to just glucose. Now, one last thing that we should explore in this process is, I already explained several slides ago how we have different molecules being converted into some of the intermediate molecules, meaning molecules that are involved in the process of gluconeogenesis. So one that I want to focus on is the Cori cycle. In the Cori cycle, specifically, is where we have lactate that is produced in muscles during the process of glycolysis. And if you remember from glycolysis, lactate is going to be produced in our body when we are under anaerobic conditions, okay? Well, what happens to that lactate? Well, it is transported into the blood, and then from blood, it gets to the liver. So what happens is that in the process of gluconeogenesis in the liver, we have lactate going back to glucose, okay? And once that glucose is made, then it can be transported back into the muscles through the blood system, okay? And as you can see, glucose can be stored as glycogens until it's needed to be degraded. You may ask yourself, why is it, or how are we able to convert lactate to pyruvate? Well, there is an enzyme that is called lactate dehydrogenase. And lactate dehydrogenase is going to take lactate, and as you can see, this is an oxidation reaction in which we can take lactate and convert it to pyruvate. And in the meantime, this also produces an NADH, which is a reduced form of a coenzyme that can later be utilized to make more energy once we get into chapter 19 and 20, where we discuss the TCA cycle and oxidative phosphorylation. As you can see on the left side of the slide, we have a diagram illustrating how does the production of lactate cycles through the body to remake glucose.